Good morning. I want to thank all of you for joining us today for this exciting event. I know that you've joined us because you're all interested to hear more about our most recently completed COVID-19 study, which I believe to be the most comprehensive animal study to date on COVID-19. So I won't take too much time here at the beginning, but I do have a few comments to set the stage. Before we get started, though, I have some logistical comments to make, which is we do have the opportunity for Q&A in this session. So there's a Q&A box. So please type your question there. We'll have time to do Q&A. And if we don't, if you are not anonymous, if you put your name in, then we can email you a response after this is over. So I want to first point out that this study would not have been possible without the generous support of so many of our donors and other contributors, many of whom are on the call today. When the world realized that COVID-19 was actually turning into a global pandemic, and scientists worldwide began racing to find ways that they could help, Texas Biomed knew immediately that it was all in based on our mission. We had the assets and the expertise to provide critical answers and resources in the fight for COVID-19, but a study of this magnitude to begin immediately required an immediate influx of capital. So I am so pleased to say that our community responded with more than 300 donors giving more than $3.5 million in a span of 7 days. Unbelievable. And the philanthropic efforts have continued as we've currently raised more than $5 million to continue our extensive COVID-19 work. A special thank goes to our board of trustees and particularly to our chair, Dr. Jamo Rubin, and our vice chair, Dr. Andy Anderson, for their leadership and critical insight during this time. This support not only provided the means by which we could get started, but it also added to the motivation of our many outstanding scientists at the Institute. And we knew the community was counting on us, and we had to deliver. That is, you were providing a vote of confidence, and we were going to get the job done. Now, I'm pleased to report 8 weeks later, we have some exciting results from our first large study. As I mentioned, I believe this to be the most comprehensive study, and you're going to hear about it in just a moment. But why is this study so important? Well, we know that animal models are a critical component of the biomedical pipeline necessary to fast track any new therapies or vaccines that come to market. We'll not have a vaccine for COVID-19 without an appropriate animal model in which to completely test safety and efficacy. Some of you have, probably most of you, have read almost every night that vaccines are already being tested in human populations. But these are largely small phase 1 clinical trials whose primary goal is to test safety. These same companies will have to test their candidate vaccines in an animal model to ensure that the vaccines work in a large population, such as being considered for COVID-19, and importantly, without any serious side effects. As we start to narrow down the candidates and discover better candidate vaccines and therapies, these too will need to be tested in validated animal studies. Animal models will also give us significant insight into the immunologic response we need to further refine vaccines as time goes on and to truly learn fundamentally what a protective immune response looks like. Importantly, Texas Biomed has a long history of conducting so-called well-documented studies required by the FDA to bring any new therapies or vaccines to market. They and our industry partners know this. This animal study is just one of nearly a dozen studies now already underway at the Institute in a number of different models. But this one is a critical first step in the process to find those needed therapies and vaccines that we all want. I'm so proud of our team, our warriors here at Texas Biomed who completed this study in such a short time. We had nearly 40 team members, an outstanding faculty group of 7 working around the clock, not only to complete this large study, but also to write the manuscript that is going to be going to a very top journal. We're currently acquiring all the necessary approvals, and it will be submitted within the next 72 hours. This too is important because it means that the research community around the world will have the opportunity to scrutinize our work to ensure that it is of the highest quality. And we're confident the study will be published in the coming weeks. So with those few opening comments, again, I want to thank you for joining us. And I'll turn this over to Dr. Deepak Kaushal, who's the director of the Southwest National Primate Research Center here at Texas Biomed, who led the team effort on this first study and is the senior author on the manuscript, and he will share with you these results. Dr. Kaushal.
Thank you very much, Larry. Good morning, everybody, and thank you for joining. I'd like to start out by thanking three different groups of individuals. As Larry mentioned, this study would not have been possible without generous and rapid support of dozens of our Texas Biomed donors and supporters. And then Larry also mentioned a group of 40 scientists, including 7 faculty members that put their labs, that stopped doing the work that they were supposed to do and put their labs to doing this sort of important work. And then finally, I'd like to thank each one of the 300 Texas Biomed staff members. Some of them are scientists that actively contributed to this work and will be recognized on the paper, but there were people providing administrative support, veterinary tech support, grant management support that were all crucial for this study to be completed. So thank you all. As Larry mentioned, this is an important announcement, primarily for the research community, because as we know right now, there are no cures or vaccines available for COVID-19, which is the defining pandemic of this era. I cannot, as a scientist, think of more worthwhile research activity at the present time than to search for answers that will have an immediate impact in combating COVID-19, both by developing therapeutics to treat diseased patients and vaccines to immunize the naive population, which by some accounts is 90 to 95% of the entire world population. So as scientists, we're always told, is your work significant if it impacts 90 to 95% of the world population? I cannot think of a more significant scientific endeavor. While scientists worldwide have been studying the macaque model for some time, here at Texas Biomed, we decided to compare SARS-CoV-2 infection in 3 different species of experimentally infected non-human primates, for all of which we have breeding colonies within the SNPRC, a component of Texas Biomed. And these were rhesus macaques, baboons, and marmosets, all of different origin, Indian origin macaques, African origin baboons, New World origin marmosets. Because as scientists, we knew that heterogeneous responses that we see in the human population to SARS-CoV-2 infection need to be studied in non-human primates and would be needed if we are to truly advance therapies and vaccines. Furthermore, we addressed whether age as a contributing factor to advanced COVID-19 disease could be modeled in non-human primates. Here are our findings in a nutshell. Our findings support that the rhesus macaque is an excellent animal model for development of vaccines against SARS-CoV-2 infection and COVID-19 disease. Over the course of the study, we completed more than just clinical observations on these animals. We also measured, using state-of-the-art techniques, exact viral loads in different compartments of these monkeys: lungs, nasal washes, oral fluids, rectal swabs, etc., etc. We also conducted state-of-the-art CT scan imaging of the lungs of these animals. And then, of course, we studied the immunology in the various tissues of these animals. And what we found was that macaques developed clinical signs of COVID-19 and also signs of lung impact, of pneumonia in the lungs. And they also had cytokine and immune responses. So macaques rapidly developed clinical signs of viral infection and inflammation. And this was coupled with evidence of viral replication early on and of moderate pneumonia in the lungs, as well as pathology in the lungs and other tissues. Longitudinal studies showed that while both young and old macaques developed early signs of COVID-19, both groups also recovered within a 2-week period. Recovery was characterized by a complex inflammatory response in the lung, particularly of myeloid cells that express a prominent interferon type 1 response. We think that these immunological findings help explain both the development of COVID-19 pneumonia and the subsequent control of this pneumonia and this disease by expression of a type 1 interferon response. So when you look at the lungs of these animals, the pneumonia is characterized by the presence of interstitial and alveolar macrophage myeloid cells. And those are the very cells that are required for control. So we speculate that the responses that we see in our rhesus macaques help clear the majority of the virus, leading to the resolution of the pneumonia and also limiting a cytokine storm. The macaques did not develop as strong COVID-19 pathology and viral shedding as did baboons. So macaques had moderate disease. Baboons, by some measures, at least two very important measures, pathology and viral shedding, had even more disease than macaques did. These results suggest that while macaques would be very good models for testing new vaccines, baboons would be good animal models for drug and therapeutics development, because drugs are meant to alleviate symptoms of disease. And so you need a model where disease is more prolonged and more intense. So different NHP species that we studied, to wrap up, recapitulated heterogeneous progression of COVID-19 with rhesus macaques and baboons developing different but quantifiable disease attributes. These are really important. We can quantify very accurately how much virus is present. We can quantify using CT scanning how much pneumonia is present in the lungs. And then we can test if vaccines lower those attributes, if drugs lower those attributes. So these attributes make these two models immediately available and essential models to test new vaccines and therapies. As Larry mentioned, defining a large animal model is a critical step in advancing COVID-19 vaccines and therapeutics. We are very excited as part of this team to have done this work and to move forward in the scientific process. Thank you again for your support. I would at this point of time like to turn it over to Dr. Joanne Turner, Vice President for Research at Texas Biomed, for some final comments.
Thank you, Dr. Kushal. Those are really interesting findings. Good morning, everyone. I think this is an incredibly important scientific announcement that we've made today. And we realize everyone's wondering when we're going to have the vaccine ready for COVID-19. And given the urgency of the situation, I think that's a really valid question. And what we really need to develop a vaccine and to do it well, so we make sure it's safe and effective, is time. And that's something we really don't have at the moment. We have a pandemic ongoing. And you'll hear 12 to 18 month timeframes for making a vaccine. And that really is an accurate timeframe. And if anything, it's an expedited timeframe. So we're trying to speed the process up as much as possible. And so this study is part of that process. We've been able to do this pilot study in a very short timeframe. It's something that normally would have taken months to years, and we managed to complete it in the shortest space of 2 months. And so we can't really underscore enough the speed in which we got this complex study completed, and that paper's ready for submission. So where does Texas Biomed go from here? We've finished a pilot. We have a lot more work to do. We're working on multiple vaccines, therapeutics, other studies to understand basic biology of COVID-19 infection. A lot of those are partnering with universities, private research institutes, and several Fortune 500 pharmaceutical companies. A lot of these started before we'd even finished the pilot. They were in line to work with us, and they've already begun. And really what makes Texas Biomed a special place is that we have this ability to combine both basic research of discovery and discovery science with the ability to do contract studies. And that allows us to work through processes quicker. We have a team of dedicated scientists that are doing regulated documentation and QC on all the studies that have come out. And that allows us to essentially push that data through to the FDA process so that it can be used in humans sooner. Now, why are we investigating multiple vaccines? Obviously, because one may not work. We don't want to put everything on one vaccine, even though it may look great. In preliminary studies, it may not work in humans. And so we have to test multiple vaccines with the hope that one of those will be effective. And then more than likely that first one coming out will be good, but perhaps not the best we could have. And so we have to keep refining that. And that's part of our process forming basic research projects, is to refine those vaccines that may work in the future even better than the ones we have now. So we need to cast a wide net, and we need to look at lots of vaccines right now and test many different vaccines with the hope that we find one or maybe more than one that's actually really effective. And we're not just doing this in non-human primates, even those are critical animal models to be able to push vaccines through to final use in humans. We have studies on our campus using rodents, so mice and guinea pigs and hamsters are the primary animals that we use, and they have a lot of value. They're really easy for us to screen vaccines very quickly. They're small, they're cheaper to use, they take up less space, and so we're able to screen vaccines and do preliminary studies before we put those into non-human primates. Some of them also have symptoms that we haven't seen in our non-human primates that let us understand the disease process more. And so some of those are basic biology studies. We have scientists on our campus now doing research, understanding an infection, trying to understand how it interrelates with heart disease and diabetes. We're looking at new diagnostic tests. All of that work's currently on our campus. So we're really confident that our researchers have and continue to make significant progress against this infection that we really have only heard about for the last 6 months. It's brand new. We're doing this because we're very collaborative, and the scientific community globally has become very collaborative. We know that this is a pressing concern that we need to fix. On our campus, it's been amazing watching everybody interact together because they really are coming together for a common cause. And as I mentioned before, vaccines aren't quick to develop. We need to do them carefully. We need to do them effectively so that they're safe when we put them into humans, but we've been able to shorten that timeline. What we've done here at Texas Biomed is part of that process. By finding the right animal model within 2 months, we now can already start vaccine screening in our animals. Really important to that endeavor was the funding that we received from our donors. So we could get working quickly. Even before we'd written grants to ask for federal funds, we have been able to start very quickly and rapidly and get this data out very quickly. So obviously those funds were essential for us. We're really thankful for those funds that allowed us to get a jump start on many others. And I think also important to mention is that in that 2-month time frame, not only did we do 3 non-human primate studies, we completed those studies successfully, and we've written the paper that's ready to come out and be published and will be submitted this week. And our scientists have been writing grants to further the research on coronaviruses and COVID-19. And it's all been ongoing on our campus when we've had about half of our staff working safely at home. And so we have not had the full workforce on our campus, that those that have been on our campus all the way through to keeping our buildings running, down to doing the actual work with the animals and the analysis, have been working incredibly hard. We have so many people working. We had a team of faculty primarily driving the studies. That's Doctors Anderson, Carrion, Giovannani, Koshal, Patterson, Martinez, Sobrino, and Torres. And they've done a great job, and they've worked with research staff, veterinarians, everybody who's been involved in compliance and animal care, everything that's allowed this study to take place and to take place so quickly. So I'm really grateful for their energy that they put into this work. They've put in many hours over and above what they would normally do. And so many people have come together for this project. And I really thank you as our audience for your time and for the donations that you gave to our institute to make this possible. Thank you. And I'm going to pass over to Corbett Christie, our Vice President for Development.
At this point, it is your time. You're on. And so we're going to do some question and answer. And to help us through that, our moderator is Lisa Cruz. She will be the voice you hear repeating your questions. And then I will be back for a wrap up after we've done the Q&A. So Lisa, you're on.
Thank you, Corbett. We have had some questions coming in, and the first question reads, for those who are being tested for having the antibodies, which appears to be very popular lately, will testing positive or negative for having antibodies for this novel coronavirus have any effect on the efficacy of the vaccine or other treatments?
So the development of antibody tests has been a cottage industry with so many companies coming in with different platforms to test whether someone has antibodies. And the assumption is that antibody production is equal to protection against COVID-19. So there are currently some problems with this. The first is that the tests overall have not been performing well enough. That is, that if somebody is told they have antibodies, that may not be true. Secondly, we do not know whether there are good antibodies and bad antibodies, which antibodies actually are protective. And third, how long is that protection? How durable is that protection? So at the current time, these tests are not at a level to be able to tell you you're protected from COVID-19. The more sensitive and specific test out there is a diagnostic test for the virus, which is using molecular biology approaches. It's a good test, and the availability continues to increase. But we know there's a time period before that test is positive where one may be infectious, and then that test may remain positive for quite a while. And we know that this virus is infectious, that is still going in our community. So at the current time, the antibody test is not predictive enough for us in a way that's sensitive and specific to give us real understanding. However, those tests are improving and ultimately may prove very useful in contact tracing and in epidemiology to understand the population as a whole. So there is some utility, but stay tuned, more needs to happen.
Thank you, Larry. This question for either Dr. Kushal or Dr. Turner: why is this primary research on animals so important toward a human vaccine?
As Larry mentioned, people are conducting some trials of vaccine candidates directly in human beings. Ideally, under normal circumstances, that's not done, and for good reason. These are, of course, not normal circumstances, and some shortcuts are being taken. But what will happen is the lead vaccine candidates will need to demonstrate immunogenicity and efficacy in robust animal models before these vaccines can be given to large number of human beings, and phase 2B and phase 3 human trials can be realistically performed. And animal models will need to have quantifiable metrics. And that's the kind of result that we've presented today, that a particular group of monkeys that are experimentally infected with a said known quantified dose of virus produces between X and Y% of pneumonia at day 3 versus day 6 versus day 12. Once these models exist, you will see a lot, once these models are publicized, you will see a lot of corporate Fortune 500 companies actually want to do a lot more, want to do these vaccine trials with us and other people who would have developed these models. Some, as Dr. Turner mentioned, are already doing so, despite the fact that our results are not publicly available at this point of time, but they know this through other channels. But unless these data exist, it would be very difficult ethically to have these vaccines delivered to 5,000 to 50,000 individuals. That is a requirement for phase 2B to phase 3 trials.
I can add a little to that. I mean, the reason why we develop vaccines is because we're trying to improve human health and increase our ability to prevent infection. And so there are 10, 15, maybe 20 vaccines that people are looking at that may potentially work against SARS coronavirus 2. We can't put all of those in humans because we don't know whether they're safe. And so if our goal is to keep the population safe, then we shouldn't be putting test articles essentially into humans. We need to put those through a model where we can first determine whether they actually have any effectiveness and don't have any really negative side effects. So that's the first screen, and that's why animal models are so important. And we often do those in small animal models because we can do them quickly because there are so many vaccines for us to test. And then the ones that look very, very good, we'll put into non-human primates as really that gold standard, as being the closest we can get to humans. And so it wouldn't be a good idea for us to just go out and test something brand new in humans because we may do harm, and that's not our goal, is to do harm to humans. Our goal is to improve human health.
Thank you both. With regard to vaccines and the timeline, Dr. Turner, you alluded to this a little during the conversation, but how realistic is the president's timeline of vaccine available by the end of the year?
I think it's challenging. Obviously, if we'd known more about this virus over the years, we could have come closer. And we do have clues from SARS-1 and MERS that help us actually develop vaccines for this specific virus. But it seems ambitious. We're trying to compress the timeline as much as we can, as we've all discussed. We're doing animal studies while phase 1 human safety studies are ongoing for some vaccines that we know actually seem quite likely to work. And so we're doing our best to shrink that down, but in reality, that's a tough call to say that we would have a vaccine not only tested for safety and for its effectiveness, but also put through the production so that we have enough to deliver out to the population. Seems very unlikely we can do that by the end of the year. But this is an incredible amount of collaboration that we're seeing right now across the scientific and pharmaceutical companies and research institutes to try and accelerate that as much as we can. We know that this is a really critical thing for us to achieve. We're doing as much as we can to shorten that timeline, but it still is a very optimistic timeline.
Great, thank you. And you mentioned that that Texas Biomed is working on multiple vaccines here. Are we able to share how many we're working on at the facility right now? Or how many candidates that we have?
We will not at this point have an exact number because we continue to be conversing with partners. So this is a very fluid situation right now. But there are several we're working on, and I think there'll be more information coming down the road in a bit. A lot of our studies are just beginning now. So we want to continue to interact with all of you as much as possible to let you know about our progress. Part of the science right now is rapid communication to the public. And I will just say that is mostly good, but sometimes all this information could be so confusing. And a lot of it that we're hearing about in the public today has not been vetted. To come back to my opening comment, it's critical that our studies go through peer review and that the work is scrutinized and we know it's proper and of high quality. Otherwise, if we're reporting on early studies or studies that haven't been scrutinized, then there's always a risk that maybe they're not as accurate. You know, we stand for high quality. Texas Biomed for decades has been doing work like this, largely related to infectious diseases, but other diseases as well. And we will continue to put all our emphasis on performing rigorous, high quality work that we internally scrutinize heavily before it's released to the public.
Thank you, Dr. Sussender. Recent news came out about Southwest Research Institute supercomputer narrowing down models for vaccine tests, the Rhodium model. How are we helping and collaborating in this initiative?
Let me just say that San Antonio is an incredible environment, a very service-minded community, the desire to really improve the human condition, something Dr. Turner emphasized, and I'm incredibly happy to be part of this community because everyone is all in and understands the importance of this. And that's likewise of our research institution partnerships that are going on. The San Antonio Partnership for Precision Therapeutics is one of the newest one, and that is really among the 4 large research institutions in the city, where the president, all of us as presidents, UTSA, UT Health, SWERI, and us, we have an ongoing relationship, particularly now with COVID-19 collaborative work. And in addition, we have this Vaccine Development Center that Dr. Turner is the executive director of. So I just want to start by saying the collaboration is essential. That's how one does big science, not incremental science. So SWERI has incredible capabilities, led by Adam Hamilton, of course, the CEO, in evaluating a large number of compounds through some of their in silico technology that they've perfected, which allows them to identify compounds that on a computer look like they may dock well in terms of neutralizing this virus and can rapidly develop these compounds. This partnership represents the ability to take that technology and then partner it with the biology opportunities in Texas Biomed. That is what you've been hearing today, our animal studies, our small animal, large animal, and even our tissue culture assays, to then do so-called throughput science to understand which of those compounds truly do have efficacy against live COVID-19. This is an ideal partnership, and there are others. This is just one example. where we're working together, taking our talents, bringing them together, and understanding the value that each of us can bring to the table.
Thank you, Dr. Schlesinger. We had a question come in from our Facebook live feed. When running the test on both macaques and baboon, was it noted in either species that they were able to mimic respiratory transmission, coughing, sneezing, or is this something that we would be focusing on later in future studies?
I can take that question. Respiratory symptoms were present in many of the animals, both in macaques and baboons, including coughing. There were other respiratory symptoms that are directly implicative of COVID-19 disease, progressive pneumonia measured by CT scans. So no pneumonia at day 0, and you infect the animals, and a day later you have some pneumonia, and that it increases over the course of time until the immune response comes in. So clearly these animals had a respiratory disease related to COVID. We haven't specifically addressed the question of aerosol or droplet transmission. That would require us to put naive animals, the ones that are not diseased or not infected, in vicinity. And there are several technical challenges as to how to do that. The labs in which these animals are, its air is actually very clean. So the entire air mass of this lab is purged out and replaced with clean air every 6 minutes, because this is a biosafety level 3, high-level biosafety lab. So if you put a diseased animal 6 feet away or 4 feet away from a naive animal, it means the process by which the air is cleaned to keep the people working there safe from all the pathogens may not allow a direct comparison of this process as it may happen in a grocery store or in a restaurant in human beings. So there are some technical challenges to doing that experiment, but still, these experiments can be done, and we haven't yet addressed this. Please keep in mind, this is a very preliminary pilot study of 3 different species. There's a lot of other things that remain to be done by us and by others.
Thank you, Dr. Kishal. Dr. Turner, I think I'll address this question to you. What is a reasonable goal of a vaccine trial? Should we expect that a vaccine offers full protection, or is it more realistic to expect a vaccine to simply improve outcomes for infected individuals? How would that change a clinical trial?
In an ideal world, we'd want full protection, and it is possible with some vaccines. Obviously, we've eradicated smallpox as a great example of an effective vaccine, and then we have other vaccines that work most of the time. And sometimes that depends on the host, right, the human host. If somebody, for instance, is older, they'll often respond less effectively to a vaccine, and it may not protect them at the same magnitude, and the flu vaccine is a good example of that. We also have to depend on every single person in the community having the vaccine, right, so that we get really good, robust coverage around our community. And so as much as we'd love it to be 100% effective, the reality is there's lots of reasons why it might not be 100%, but we don't have to receive exactly 100% to get really good protection in our community. We can be a little bit less than that, but not much more. So everyone who can get vaccinated should get vaccinated, because they're protecting the individuals that are unable to generate protective immunity for themselves. We may also have vaccines that reduce some symptoms, and that's also good, right? So if we can maybe not necessarily prevent infection, but stop somebody getting so sick that they end up in the emergency room or in an ICU, then that's also a really good vaccine. And so the first ones we bring out may do some of what we need, but not everything. And then we're going to keep refining them until we get that vaccine that really covers everything that we want and gives us really, really good protection for the whole population.
Thank you, Dr. Turner. Dr. Kishal, one of the aspects of this study and what made it so comprehensive was the idea that we looked at both young and aged animals. And so did older animals display greater vulnerability to the disease, or did we see any differences?
This is a great question, and the answer is no. That was the premise for doing at least this part of the study, is whether we would see greater progression of infection in aged animals, older animals, and we did not see that. And there could be several reasons for that. Of course, you can doubt our model, but it's unlikely given that we have measurable attributes of COVID-19 disease. We do think that these models are quite robust, and others think that too. We think the fact that we did not see greater disease in older rhesus macaques as compared to younger rhesus macaques is an attribute to the fact in human beings, age is not the defining factor. It's other things that happen as a result of age. Immune-related inflammation with aging, and diabetes and cardiovascular disease and other things that develop with aging that happens more so, in a more defined manner in human beings than it does in older macaques. That those are the comorbid conditions that lead to advanced COVID disease and not aging per se. We have not conducted this, we have not completed these studies in baboons. Remember, baboons had more disease than monkeys. We've only completed the young baboons part. The older baboon part is being completed. That data is still being analyzed. It's quite possible that we'll find some differences between younger and older baboons in terms of aging, but at least in rhesus macaques, we did not.
Great, thank you. We have a question about virus evolution. Will that be a problem for vaccines in terms of development, and will this be like a flu vaccine where we need one every year?
So let me address that. Let me start with good news. Viruses are evolving all the time. In fact, one of the most ambitious viruses that evolved during an outbreak is Ebola virus, which we recently had an outbreak which is under control, in part related to studies completed at Texas Biomed. But here's the good news. Almost always, the mutations acquired or variants that are acquired in the virus do not fundamentally change the ability to cause disease or the way in which disease develops. So by and large, the virus behaves the same way, even though we know COVID-19 is evolving. Does that mean it could not fundamentally change? So for example, we know that there are these proteins on our host cells, and that's part of the evolution of COVID-19 was the ability to recognize those proteins as a lock and key type mechanism to begin this whole disease process, and that's part of its new infectiousness compared to the other coronaviruses. Is it possible that a virus acquire a big so-called deletion or change that makes it fundamentally change? It's always possible. That's why we need to continue to sequence viruses that are circulating in the population and know whether those changes occur. But it's infrequent. Now, the latest information we're hearing about, and the question we don't have the answer to, is will COVID-19 end up being another global endemic virus after the pandemic? That is it going to just go away, or is it now going to be another one of our respiratory viruses that we need to be very concerned about? That we're uncertain of. But that will also play into whether a virus evolution is important or not. Let me finish by saying that the influenza vaccine, a critical vaccine because of the vulnerability of those in the older population, is really at best about 40% effective. Whereas, for example, the measles vaccine is over 90% effective, okay? By the way, we need to develop these vaccines and then take them. Let me just emphasize the safety feature that's been emphasized in these animal studies, because we do have some people who are hesitant about vaccines. We must ensure the public that these vaccines are safe. But we need to know whether COVID-19, whether it will be with us going forward or not, and that becomes really critical. But with the influenza and maybe with coronavirus or COVID-19, rather than proteins that do change, like the coat of influenza, we need to develop these so-called universal vaccines that recognize core or parts of the virus that don't change, okay? And Dr. Martina Sabrita, who has recently joined us, plays a critical role in our virus team at Texas Biomed, came here with specific technology to develop a strategy for a universal influenza vaccine. Do I think there could be a universal coronavirus vaccine? I do believe we can get to that point, so that even with some of the evolution that occurs, the vaccines will continue to be effective. But those are the kinds of studies that need to continue. We have a long way to go.
Thank you. I'm going to ask the final two questions. First question, Dr. Schlesinger, can you comment a little on the level of private industry interest in the animal model study? Very briefly, please.
There is a lot of interest for the reasons that have all been stated today. industry is coming forward as well. The partnership between industry, research institutes like Texas Biomed, academia, everybody's all in. This is very competitive, but it's absolutely necessary that we work together. Industry understands that well-documented studies, those that the FDA requires, that is quantitative measures, become valuable for any new diagnostic test, therapy, or vaccine to make it to the market. That level of rigor needs to be there, and these models are controlled so we can test a lot. Human trials are extremely important, but they cannot do the same thing as these controlled studies in the laboratory can do. So the partnerships are very strong, and we welcome them and have a long history of working in the private sector. We, of course, are also a not-for-profit freestanding research institute. And I think that what is unique is something Dr. Turner said, I'll end there, that we're a hybrid organization. We're unique in so many ways. We have outstanding fundamental science because fundamental science is the bedrock of any future diagnostic therapy or vaccine. And we also have a track record of working with industry partners under these contract science rubric, that is the carefully well-documented study. We have experience with that, and that's unique for us, so we can do the fundamental science and then the science that's closer to the clinic. And so that becomes a critical arm of our institute.
Thank you, Dr. Schlesinger. And for those questions that we didn't get to, as long as we have your name, we'll make sure that we type answers and get those answers out to you via email. The last question actually is for you, Corbett. Given that the government funds much of this, how can donors help with this effort?
Oh, my goodness. Thank you. Thank you, Lisa. And thank you for the question. Independent biomedical research institutes thrive on private support. It's the way we built most of our campus, most of our labs, and enables us to cut through a lot of the red tape. This example of this very study is perfect, and it is private support that comes directly to this institute that's going to reposition our labs, our biosafety level 3 labs, so that we can do the kinds of studies that we need to do. We've got to beef up our animal space so that we can do research on more animals. And so this is a very rational plan that we go forward. We don't leave any stone unturned. We do ask for federal or state or even other types of support that's more public, but it is private support that's the bedrock of this institution. So I think that would be the way I would answer that question. And I guess now, Lisa, it is time for me to do the wrap on this. I'm the only non-scientist on this panel, and I'm going to try and bring this home to something that makes a lot of sense to me, but also makes, I hope, a lot of sense to you. And this, what's in this for me? Have you ever, at my age, I wake up in the middle of the night, and it's dark, and I've got to take steps. And let me tell you, those steps are not very confident because of the darkness, and that unknown out there is overwhelming. But isn't it miraculous when you have a beam of light to guide your steps? And that light is like a miracle. Well, the world awoke just a few months ago at the descending veil of darkness, this chaos and this fear, all the result of an unknown virus that was identified at the time and still today only by letter and number. This darkness created fear, and we need light and knowledge to see our way out of this. I believe that we'll emerge from these fearful times, but how? It will be our knowing. It will be the knowledge that comes from work like we're doing at Texas Biomed in research that will lead us to the light of the known. And out of this fear, we will gain confidence. We move to confidence. And from darkness, we will move to light. And so what you've heard today is one strong beam of light. The project that Drs. Schlesinger, Koschel, Turner, and those other heroic 40 or so people who work 24/7 to push this project through are true heroes, and they are going to help lead us to that light. We have learned a lot about COVID-19, and we'll bring light to the darkness. Our founder, Tom Slick, put us in this place. He knew that this was the way that we could power human progress through scientific research. So we've done this together. We'll do it together. And I'll tell you, we need your prayers. We need your support, your financial support, and we need you to believe that we will move from a period of darkness to a period of light. And it'll be by knowledge, and it is science that will lead us, and it's research that will lead us to this. Together, we will overcome these fearful times. So thank you for coming out today, and we hope that you will continue to stay in touch. We're going to be good communicators, and we will be in touch soon. Back to you, Larry.
Oh, okay. Well, thank you, Corbett. That said it all. I'm a relatively new San Antonian. It's my 3rd year anniversary here. I came here because I saw something very unique, not only in San Antonio, but for the state in the country, and that is Texas Biomedical Research Institute. This is truly outstanding environment for learning, our educational programs, but it's also an outstanding environment for discovery-based science and for our ability to really make a difference in the world. I wake up in the morning as a physician scientist thinking about how can we do better, and right now we're in the midst of a pandemic. For me, this is not the first time I've experienced outbreaks like this, and I know it's not if, but when, but we truly need to become more aware, more knowledgeable, and think about the next one and be prepared. Texas Biomed's goal, its mission, its vision is to lead the nation in being prepared. Our mission is to protect you and the global community from the threat of infectious diseases. And every day, our scientists, our staff, our facilities are built to try to help save a human life through the problem of these infections. Like Corbett said, this is a team effort. We need your support. We truly need your support, and we aim to deliver. So thank you. Appreciate the time that you all took. We had nearly 100 people on this call from various walks. I hope we've been able to answer questions and address some issues. I know there are more, and we're always available to answer those, so please reach out. Thank you.